Hey, bacteria. Let's do it. All right. So you should have your science book open to page 78. If you don't, go get it because you're going to have it. You got to be prepared. And you're going to need a pencil. Go ask your mom for a pencil. All right, good. So now you're watching the video. You got a pencil. What I want you to do is you pause right now after you listen to me. You're going to go to YouTube and you're going to search in how does bacteria make people sick. You're going to watch that so you can have a base understanding about bacteria, how it makes you sick. In order to succeed in this, you kind of need some background information. It's easier for you to do it at home. You get the background information, then you'll watch this, and then you'll be able to succeed. Very good. So I assume since you're still watching, you went and watched that YouTube video, which is what you should have done. I'm going to use a combination of media. I've got my book in front of me here and I also have my iPad. I will direct you as to which one I'm going to use. So I'm on page 78 in your book, page 79, the vocab terms, which are bacteria, archaea, binary fission, virus, and host. You guys need to have all 5 of those on a piece of notebook paper turned into Miss Jones's desk tomorrow. So that's what you're responsible for. And completing pages 79 through 85. Yeah. You're going to do those pages. You'll come in tomorrow. We'll have some awesome work. We're dealing with like maybe some Play-Doh or Wiki Sticks or some sand. And we're going to be making some bacteria in our classroom. So let's go ahead and get started. So you're going to follow along with me in the iPad. Then you're going to be able to work on the pages in your book. I'm not going to read the book. I'm not going to go through each page by page. But just by following along, you'll be able to succeed with that. You'll come in tomorrow. It'll be awesome. All right. So bacteria, what is it? It's a very, very small thing. You can't see it with the naked eye. So you can't walk down the street and trip over a big pile of bacteria. All right. You're not going to leave bacteria on the floor. Mom said you pick up your bacteria, throw it in the hamper. That just doesn't work like that. It's microscopic. It's very, very small because it's single celled. All right. When we're working with bacteria, there's 2 different types of cells. The type of cell that bacteria are prokaryotes. Prokaryotes they have no nucleus. They have no membrane-bound organelles. So this one over here is a prokaryote. There's no nucleus in there. When I look at a eukaryote, which is the type of cells that plants and animals are, I see a nucleus. And these little guys in here are organelles, and there's a lot of functions going on. A prokaryote does not. It has what's known as free DNA. It's just kind of popping around in there. So eukaryote, prokaryote. We're dealing with prokaryotes this lesson. All right. So a couple things about bacteria. They're single-celled. We're going to learn about their shapes. We're going to learn that they live everywhere, that they divide with binary fission, and how they exchange DNA. So we'll cover each one of those, and I think it's in order in your book. Hopefully it is. All right. What I want you guys to see now is the basic shapes of a bacteria. And here's what a basic shape of a bacteria is. When you think of bacteria, that's what you see. When you look in your book, this is what you'll see. You'll see that the basic shape is a capsule. So it looks like a pill, a capsule. And inside, there is called nucleode. It's circular DNA. It's right in there. They move around by bacterioflagellum. There's all kinds of membrane and cytoplasms, and you'll be able to look in there and see that. So the basic shape of a bacteria, all right? Very, very microscopic. Like I said, single-celled. The free DNA in here, the fact that there's no nucleus, is going to come into play big time when it's time to divide these guys. Let's talk about their shapes. You're going to learn about the shapes of the bacteria, and they come in really, really awesome shapes. And then you guys are going to see this right now. So there's 4 different main types. This first one is called a spirillum, and look, it's like a chain of diamonds. Real tiny and real organized, really straight line. When in class we'll learn about the different types of bacteria and what they are. The next one I have is bacillus, and it's kind of like a curlicue, kind of wacky looking. So this guy would be like the straight and narrow cousin, and this guy's the wacky one that goes off and does his own thing. So there's a big difference there. We have Coccus, which, you know, we can see what those are. They're just like spheres, and they look like they fall in a pretty good order. And then we have Vibrio, and Vibrio, they're kind of hooks, and we'll learn about all the different types of bacteria there. All right. The next thing we have to talk about is how they divide. Bacteria, since they're a single cell organism, it's really easy for them to divide. They use a thing called binary fission. We're going to learn about that right now. Binary fission is they are simply multiplying their DNA, and then they are splitting and then moving on. And they can do this in a pretty rapid state. So we're going to go through each of the phases right here. So binary fission, this is what we're talking about. First step is you see that there's a single bacteria cell. And the illustration is a nucleus, but we'll call it its DNA. All right, so the DNA is just simply right there. And when it wants to start dividing, it divides the DNA inside first. So it copies itself, and then it starts to split. And then step 3, we see that there's a little pinch, and it's starting to cut itself apart. And then it's more divided, and bam, all of a sudden we have 2 identical bacteria cells. And those can cause a whole host of issues because they divide really fast, and then they can go and then make you sick. All right. The last thing we're going to look at on page 85 is how they exchange DNA. So when it's time to exchange that DNA, after they're done making copies, then it's time to go to work. Well, how do they do that? Well, there's 3 different types. One's called transformation, one's called transduction, and the other one on your page 85 in your book that you have to finish, it's called conjugation. And that's making a bridge. And I'm going to show you an illustration with that right now. So for transformation, all it is the bacteria cell releases its DNA and it goes into a host cell and simply transforms that DNA of that host cell into the bacteria cell. Pretty cool. The next one is called transduction, where the bacteria or the virus attaches itself to its cell, to the host cell, and it injects its DNA into the cell and then takes over the cell like that. The last one is called conjugation. And what you have is a bacteria cell floating around, and it actually stretches out and attaches a bridge to another cell. When this bridge is complete, the bacteria cell injects its DNA into the new cell and then closes the bridge, and then it goes away. So